Stanford University.
It is actually true that a number of us work closely together here at Stanford, and in addition to Carl, Sam Strober, who just spoke to you, and Rob Negrin, are all collaborators in many of the work that, much of the work that our group is involved in, and it is a pleasure. I was asked to speak about dendritic cell-based immunotherapy, which I will try to do. Although this is still an experimental field and I will emphasize a lot of experimental aspects, I will move fairly quickly through a number of slides, and my apologies for having to go as fast as I will. There are today thought of two principal subpopulations or subsets or sublineages of dendritic cells. The myeloid, so-called myeloid dendritic cells, which can be derived from monocytes, which is a very common type of circulating precursor cell. And they're famous for capturing, processing, and presenting antigens to T cells and stimulating an immune response. They also activate natural killer cells or NK cells and B cells. These are the antibody-producing cells, and I think these are cells that are involved in innate immunity. The other type of dendritic cell subset about which I won't speak today, other than to describe them briefly, are the plasmacytoid dendritic cells. Their ontogeny is less clear, although our data and the data of others suggest that they are also derived principally from a myeloid progenitor in the bone marrow. That's why we call these myeloid, by the way, is that the progenitor cell that gives rise to these dendritic cells are myeloid dendritic cells in the bone marrow. Now, plasmacytoid dendritic cells are best known for being the principal source of interferon, type 1 interferon in your body, and that's a very important naturally occurring antiviral protein that is secreted by a variety of cells in small amounts, but this cell type secretes interferon alpha in enormous amounts, and it's the principal way that we protect ourselves against viral infection. Now, very recently a paper appeared suggesting that these cells also play a very important role in immune tolerance that occurs when you eat food. So when you eat proteins, we don't generally develop an immune response to the proteins we eat, whether it's from meat or eggs or milk or whatever. And the mechanism involved in protecting us against some sort of immune reaction against foods is unknown. But recently it's been suggested that plasmacytoid dendritic cells may play a critical role. But we're going to drop that subject and go straight to these classical dendritic cells or myeloid dendritic cells. They're very efficient at taking up antigens or substances from the environment, processing them, and then presenting them to other cellular members of the immune system. There are a number of molecules that they uniquely express and secrete that aid them in this process. I've identified a few of those molecules on this slide. This is what dendritic cells look like. They're present in most of our tissues, and you can see why they get their name from these finger-like processes. These cells reside in a variety of tissues where they will stay for a very long time, perhaps the entire life of the host, waiting to see if something evil or threatening comes along, which they will then react to and take up, process, and then deliver into the lymphoid organs to initiate an immune response. So this is how that process works. I've depicted here schematically dendritic cell precursors or progenitors residing in the bone marrow here as a stem cell. They then, once some of these stem cells have decided to become dendritic cells, they leave the bone marrow through the bloodstream, through the circulation, and they make their way to some particular tissue. In this case, we're illustrating the point through skin. So in this case, the dendritic cells are migrating, the precursors are migrating to skin, and then they're populating the skin. There are two types of dendritic cells in the skin. The superficial ones are called Langerhans cells, and the